Hello, this is Dr. Mathangi, Associate Professor, Department of Biochemistry. Today's session is about amino acid metabolism with a special focus on urea cycle. Every living organism excretes nitrogenous waste. There are three forms in which nitrogenous wastes are excreted from the body of any living organism, based on which we classify organisms into amonotelic, uricotelic and ureotelic. Well, human beings are ureotelic, that means the principal nitrogenous waste which is excreted from our body is urea. Urea is the end product that is produced by the catabolism of a protein. Before moving into urea cycle, let us see a brief overview about the metabolism of amino acid. Amino acid metabolism involves amino acid catabolism as well as anabolism. Anabolism, here we mostly focus on the exogenous source of amino acid, which comes from dietary protein. The dietary protein, when they are digested, they are broken down to the stage of amino acids. With respect to catabolism, tissue proteins, they undergo degradation periodically, and these degraded proteins in turn gets pooled as amino acids. Together, from the exogenous dietary source and the endogenous breakdown of tissue protein constitutes to something called as amino acid pool, which is an imaginary source of all the free amino acids existing in every cell. So, the total concentration of free amino acids that are existing throughout the body is assumed to be 100 grams. So, on the 100 grams of amino acid is actually constituted by the catabolism of the tissue protein as well as the exogenous source which comes from the dietary protein. This amino acid pool is further used as a source to synthesize various proteins which are needed for the normal functioning of the cell. That includes the tissue proteins. Nearly 14% of the amino acid pool goes into making tissue protein, plasma proteins which will be 7%, and some free amino acids are always maintained in circulation which goes to various tissue and organ system, and that constitutes about 22%, and the majority of the free amino acid gets broken down into keto acid and ammonia. This keto acid is again recycled as intermediate for various metabolic pathway, also gets converted into certain non-essential amino acid, and this ammonia, which is one of the breakdown product of amino acid, is what we are going to discuss today in detail in urea cycle, because this ammonia constitutes about 57% of the breakdown product of an amino acid. Ammonia in free form in any living form is toxic. The normal level of free ammonia in adult should be somewhere around 30 micromolars per liter. An increase in the concentration of ammonium ion to 100 micromole per liter, it leads to changes in consciousness, and a blood ammonia level over 200 micromole per liter is associated with coma and convulsions. Let us look into the catabolism of amino acid in detail. Catabolism of amino acid involves 4 stages. The first stage is called as transamination, the second one is deamination, then the transport of amino acid, and the fourth stage is urea cycle, where the liver detoxifies the ammonia in the form of urea. When ammonia is converted to urea, it is non-toxic, and it could be easily excreted by the kidneys as it is more soluble in water. So, let us go into the first step in the catabolism of amino acid, that is transamination. As the name indicates, there is going to be transfer of an amino group. From where to where? From an amino acid to a keto acid. So, we need 2 substrates for this reaction. One is the amino acid, which is going to undergo catabolism, and the keto acid. So, the amino group from the amino acid will get transferred to the keto acid in the presence of an enzyme called as transaminase. As we all know, most of the enzymes require a coenzyme for their effective functioning, and here the coenzyme is pyridoxal phosphate, which is a vitamin B6 derivative. So, the actual acceptor of the amino group in this reaction is a pyridoxal phosphate, and it gets converted into pyridoxamine phosphate, and all these reactions are reversible. So, this reaction predominantly takes place in liver, kidney, heart and brain, as these 4 organs have adequate amount of the transaminase enzyme. Let us look into the reaction of transamination, and we will be discussing about 2 transaminases which are predominantly of larger clinical significance. So once transamination is done, the end products are keto acid and ammonia. Here the keto acid will be used up for various metabolic reactions. They could act as intermediate for TCA cycle, they could be used for the synthesis of purines, pyrimidines, etc. And this ammonia, which is again formed, will be transported in the form of glutamate and glutamine. So this glutamate and glutamine will enter the liver and it gets detoxified. So at a given point of time, there are two transaminases which are chiefly functional in most of the organ system. They are SGOT and SGPT, which are collectively commonly known in modern terms as AST and ALT. Let us see the equation which represents transamination by AST and ALT. So look at the first reaction. So here we start with aspartate. The keto acid that is involved in this reaction is alpha ketoglutarate, and the product formed is oxaloacetate and newer amino acid, that is glutamate. Invariably, most of the transamination reaction ends up producing either alanine or glutamate, and glutamate will be converted to glutamine. Alanine will be mostly exchanged between muscle cells, brain cells, and this glutamate will be excreted through the liver and in the form of urea. So let us look into the reaction in detail. The first reaction, it talks about the transfer of amino group from the L-aspartate to the keto acid. So there should be a keto acid as an acceptor. So a newer keto acid and a newer amino acid is formed. So what is the basic reaction here is, anyway we end up getting a newer amino acid. So where is transamination? So the transamination is done, but when you look carefully, the newer amino acid formed is always glutamate, and that gets transported and the ammonia group gets detoxified. So the second reaction, it shows alanine transaminase, where we start with alanine. There is an acceptor again, which is a keto acid. The new amino acid form, again you can note here it is glutamate, and a keto acid is being formed, which is pyruvate. So if we see about the transamination reaction in detail, the transaminases enzymes are also called as aminotransferases. When you are asked to write the general equation just mentioned, amino acid 1 reacts with keto acid 1 to give amino acid 2 and keto acid 2, just to show the variation between the reacting amino acid and the product, which is a new amino acid being formed, invariably glutamate. And pyridoxal phosphate is the coenzyme needed to carry out this reaction. So why transamination? There is transfer of amino group, but there is no free ammonia, thereby it is non-toxic. This reaction is said to be reversible. Sometimes you might get a question whether it is an anabolic reaction or catabolic. It is both. It is catabolic with respect to the breakdown of amino acid. However, a new amino acid is being formed, so it could also be anabolic. It helps in the synthesis of non-essential amino acids. So lysine, threonine, proline and hydroxyproline do not undergo transamination, they directly undergo deamination, so they are exception to this rule. Alpha amino acids and delta amino acids both undergo transamination, and with respect to transaminases, we talk about AST and ALT, that is SGOT and SGPT. Elevation of SGOT is seen in acute myocardial infarction, and SGPT elevation is seen in liver diseases, and therefore these two enzymes are also of greater clinical significance. So this reaction, it shows transamination in detail, where you see the conversion of alanine into pyruvate, and the keto acid which accepts the amino group from alanine gets converted into glutamate. So glutamate is a collecting point of most of the transaminase reaction. So this reaction again shows glutamate as the collecting point of transamination. So what happens to this glutamate? This glutamate can further accept ammonia group, also helps in buffering, and it gets converted into a derivative called as glutamine, and this reaction is catalyzed by the enzyme glutamine synthetase, which is present in liver. So the end product of transamination is glutamate, but glutamate could further be converted into glutamine. Now let us look into the various keto acids produced as a result of transamination. So the keto acids predominantly produced are pyruvate acetyl-CoA, which gets converted into amino acids such as alanine, glycine, cysteine, serine and threonine. And they actually enter, either way, when amino acid gets converted into pyruvate or acetyl-CoA, they enter as an intermediate into the TCA cycle. The same way phenylalanine, tyrosine, tryptophan, leucine, lysine, isoleucine, they mostly get converted into the keto acid acetoacetate, which again enters into the TCA cycle. Asparagine and aspartic acid gets converted into the keto acid oxaloacetate. Histidine, arginine, proline, etc. gets converted into alpha ketoglutarate, and methionine, valine, isoleucine, threonine gets converted into succinyl CoA. And if we notice carefully into the keto acids, they are all intermediates of TCA cycle. Some also enter into the synthesis of fatty acid. Going to the second step in catabolism of amino acid, we talk about deamination. Only liver mitochondria contain the enzyme for deamination, which is called as glutamate or dehydrogenase, and it deaminates glutamate into alpha ketoglutarate and ammonia. For this function, NAD is used as a coenzyme, and glutamate dehydrogenase is an allosteric enzyme, and therefore it undergoes allosteric regulation. It is activated by ADP and it is inhibited by GTP. Deamination, specifically we talk about only one deaminase, that is glutamate dehydrogenase. Why? Because you have to recall that most of the amino acids following transamination are converted either into glutamate or into glutamine. So transamination, deamination could also take place simultaneously for certain amino acids, and we define that process as trans-deamination. So this figure, it shows the equation the schematic representation of oxidation of glutamate by glutamate or dehydrogenase. L-glutamate gets converted into alpha ketoglutarate in the presence of glutamate dehydrogenase. You can see the change in the structure where the amino group is removed and it is released as ammonium ion. So this slide, it shows both transamination as well as deamination. Again, to recall, remember, most of the amino acids, when they undergo transamination, they get converted into glutamate, and this glutamate is upon reaching the liver undergoes deamination to release ammonium ion. Now we are going into the most important aspect of amino acid metabolism, that is urea cycle. Urea cycle is the most repeatedly asked question in undergraduate examinations and is of greater significance even in theory viva. So urea cycle is a cyclic process which happens in the liver. It primarily is associated with detoxification of ammonia in the form of a nitrogenous compound called as urea. So and this nitrogenous product urea is mainly excreted through the kidneys into the urine. So as I told you already, ammonia is very toxic to human beings when it is available freely in circulation. Therefore, it is converted to urea, which is non-toxic and very soluble and easily excreted by the kidneys. So every day a healthy adult excretes about 30 grams of urea through urine, and this accounts to 90% of the total nitrogenous excretory product. So urea is formed in the urea cycle from the ammonium ion, carbon dioxide, and the nitrogen in the urea comes from aspartic acid, which is another amino acid. Urea cycle predominantly occurs in liver. Urea cycle is the first metabolic cycle to be deciphered. It is also called as Krebs-Hanslight cycle. So there are 5 steps involved in urea cycle. The first 3 steps happens in cytosol and the last 2 steps happens in the mitochondria, and this process is compartmentalized in the liver hepatocyte. In total, there are 4 ATPs utilized for the synthesis of one molecule of urea. The process is irreversible and urea cycle is called the end product of a protein catabolism. So the first step in urea cycle is combination of carbon dioxide and ammonium ion to form carbamoyl phosphate catalyzed by the enzyme carbamoyl phosphate synthase 1. Carbamoyl phosphate synthase 1 is a rate limiting enzyme and it is regulated allosterically. The allosteric regulator of this enzyme is N-acetyl glucosamine, which is an activator, and we will see the regulation part later. So when carbon dioxide reacts with ammonia, it forms carbamoyl phosphate, which in turn gets converted into citrulline by combining with an amino acid called as ornithine. So the enzyme that catalyzes this reaction is ornithine transcarbamylase. So carbamoyl phosphate in the second step gets converted into citrulline. So citrulline is getting converted into arginosuccinate by combining with aspartate as mentioned earlier, and the enzyme that catalyzes this reaction is arginosuccinate synthetase. Arginosuccinate is then converted into argine by arginosuccinase enzyme, and arginine is converted to ornithine by the action of the enzyme arginase. So this is the step where urea is formed as a byproduct. So this entire cycle starts with ornithine, and ornithine is released at the end of the cycle, and therefore this process is called as urea cycle. And this reaction, as you can see, the entire cycle happens partly in the cytosol. The first 2 steps happening in the cytosol, and the last 3 steps happens in the mitochondria. There is clear compartmentalization. Regulation of urea cycle. As I told you, the key enzyme that is regulated is the first enzyme CPS1. It is allosterically regulated by N-acetyl glutamate. Let us also see various inborn errors associated with urea formation and the enzymes involved and the characteristic clinical presentation. The first one is hyperammonemia type 1, where the first enzyme CPS1 is defective. As a result, the detoxification of ammonia becomes difficult, and the patient will experience increased serum levels of ammonia, which accompanied by severe mental retardation. The second type of defect is hyperammonemia type 2, which is caused by the defective enzyme ornithine transcarbamylase. Again, there will be difficulty in detoxifying ammonia, and there will be increased levels of ammonia as well as glutamate in the serum. The third type of defect associated with urea cycle is citrullinemia, where the enzyme that is involved is arginosuccinate synthetase. The enzyme is defective, and it is characterized by increased amount of ammonia as well as citrulline in the blood. The fourth type is arginosuccinic acid urea because of the defective enzyme arginosuccinase. So there will be increased production of arginosuccinate in blood and also its excretion in the urine. The last disorder associated with urea cycle is hyperargininemia. The defective enzyme here is arginase, and therefore there will be increased amount of arginine in blood, CSF as well as urine. Let us now understand the significance of blood urea and its clinical importance. Blood urea is the major nitrogenous waste which is produced by human body excreted through kidneys. Blood urea is also an indicator of renal function. Uremia is a condition where there is increased urea in blood because of the inability of urea clearance by the kidneys due to various factors. Azotemia is a term which denotes increased urea as well as other nitrogenous waste in blood without a renal disease. Now let us move on to the non-protein nitrogenous compound. So blood contains non-protein nitrogenous compounds. That means other than proteins and peptides, the nitrogen containing compounds are called as non-protein nitrogenous compound. The principal non-protein nitrogenous compound that will be discussed are urea, uric acid, ammonia and amino acids. While we discussed the free circulation of amino acids earlier in this lecture with respect to amino acid pool, we have to understand that many amino acids circulate freely in blood, and they will be utilized by the various organ systems of our body as they act as intermediates for metabolic reaction. Also, many biochemically important products are derived from a single amino acid. With respect to urea, we have now seen how ammonia is detoxified as urea, how it is entering the plasma and how it is cleared through the kidney. Next is uric acid. Uric acid is again a non-protein nitrogenous waste which is predominantly produced by the catabolism of purine nucleotides. So all these non-protein nitrogenous compounds which are available in plasma and also eventually excreted, they have implications in clinical biochemistry. So this tabular column, it discusses about the non-protein nitrogenous compound that are normally present in blood, and what are the clinical significance or the biochemical significance when the air levels are altered. First, talking about free amino acids. The source of the free amino acids are protein, as we have discussed already by the catabolism of protein. The clinical and biochemical significance includes the amino acids which are circulating in free form in blood. Their levels are altered in liver disease, renal disease and in inborn errors of amino acid metabolism. With respect to urea, the source of urea is ammonia, and when there is an increased level of urea in blood, it increases liver disease as well as renal disease. Creatinine, the source of creatinine is creatine, and increased amount of creatinine in blood indicates renal disease. Uric acid, the source of uric acid is purine nucleotide, and increased uric acid, it shows excessive cell lysis or any disorder associated with purine metabolism. So among the non-protein nitrogenous compounds, we have discussed about urea. Next we will move on to creatinine. Creatinine is again a non-protein nitrogenous compound produced by the breakdown of creatine in muscle. These two terms, creatine and creatinine, are mostly confusing. So kindly remember, creatine is an amino acid which is normally present in muscle cell and it helps in energy production. When it breaks down, the metabolic derivative of creatine is creatinine, which is a metabolic waste, and it is removed through the kidney. Creatinine is found in serum, plasma, and urine, and it is completely filtered during glomerular filtration, and urine formation. And therefore, creatinine clearance is used as a characteristic test to assess renal function. Creatinine is of endogenous origin and it is not of exogenous origin, that is, it does not come to the body through food or any other source. And as it is a breakdown product of creatine, which is a source of energy, which acts as a source of energy in muscle, and creatine phosphate level reflects and its breakdown into creatinine reflects muscle mass. So this image, it shows how creatine is converted into phosphocreatine or creatine phosphate. Creatine phosphate is the main high energy phosphate that is stored in the muscle. In a normal resting muscle, creatine phosphate will be predominate, but when there is vigorous muscular exercise, when there is need of acute energy, there is an enzyme called as creatine kinase which will convert this creatine phosphate into creatine, and the phosphate that is liberated will be used to produce ATP from ADP, kind of substrate level phosphorylation, which will help the muscle cell meet the energy need and help in contraction. So this flow chart again shows the interconversion between creatine phosphate and creatine, which is accompanied with the conversion of ADP into ATP that will release quick energy for the muscle cells to perform, especially during muscular exercise. Now going to the parent compound, that is creatine, from which this creatinine is derived. So this creatine is a naturally occurring amine. It is synthesized in liver and kidney by two methods. One is transamination, utilizing two amino acids, arginine and glycine. The other is methylation of guanidine acetic acid. These are the two methods by which creatine is indigenously produced in the body. Going back to creatinine, which is a derivative of this creatine, when a renal disorder is suspected, normally the ratio of BUN, that is blood urea nitrogen, to serum creatinine is useful in diagnosing and locating the problem. The normal ratio of this blood urea nitrogen to creatine should be 10 to 20 milligram per deciliter of BUN to 1 milligram per deciliter of creatinine, and this is of course the normal level of blood urea nitrogen and creatinine in blood. The normal level of blood urea should be between 10 to 20 mg per dl, and creatinine should always be 1 mg per deciliter of blood in a healthy individual. When the ratio between this blood urea nitrogen and creatinine is not 20:1, that is when it is more than 20:1, it suggests a pre-renal disease. When it is less than 20:1, again it is an indicative of renal disease. Both are should be taken into consideration. Creatinine is measured using Jaffe's method colorimetrically. It could also be measured by HPLC. Creatinine measurement by Jaffe's method is actually more commonly used colorimetric technique to estimate the amount of creatinine present in blood. So in this video lesson, we have seen in detail about amino acid, its catabolism, what contributes to amino acid pool, and what happens to the free amino acids present in the amino acid pool. Followed by that, we have discussed the catabolism of individual amino acid, which involves 4 steps: transamination, deamination, urea cycle, transport of the ammonia to the liver, and urea cycle. Transamination, we discussed about transaminases and the two key transaminases that are of clinical significance, AST and ALT, with the complete equation. We discussed about how glutamate and alanine are formed as a result of transamination and the significance, how glutamate is converted into glutamine and which helps in non-toxic transport of ammonia. Also, we discussed the various keto acids produced as the byproduct of transamination and how they get into TCA cycle as a metabolic intermediate. The second step in catabolism was deamination, specifically with glutamate dehydrogenase, and how ammonium ion is released. The third step is urea cycle. We saw the 5 steps in urea cycle, how is it regulated, its energetics, and also the disorders associated with the urea cycle. And adding on, we also discussed about the non-protein nitrogenous compound other than urea, and we have seen a brief introduction into creatinine, which is a waste product excreted by the kidney, and what is the significance of creatine phosphate. Thank you for your patient listening. If you have any queries, please write to us. Thank you.